Good morning and welcome to the POOLBEG PHARMA PLC investor presentation. Throughout this recorded presentation, investors will be in listen-only mode. Questions are encouraged, can be submitted at any time via the Q&A tab situated in the right-hand corner of your screen. Just click Q&A, scroll to the bottom, type your question and press send. The company may not be in a position to answer every question received during the meeting itself. However, the company review all questions submitted today and will publish responses where appropriate to do so. Before we begin, we'd like to submit the following poll. I'd now like to hand you over to Jeremy Skillington, CEO. Good morning.
Good morning, and thank you for that introduction, and thank you for everybody for joining us this morning on the back of some very exciting news from POOLBEG Pharma around the results, the outcomes from our LPS challenge study, which again very happy to present today. I'm joined by colleagues David English, who heads up our business development group, and Professor Derek Gilroy, from Professor of Immunology at University College London. And the plan of the format today is I'll just do some general introduction, some kind of high-level overview of the data that we've, as I announced this morning in the RNS, positive results from POOLB 001 and the LPS human challenge trial. Again, very exciting time for POOLBEG, a great day for the company and ultimately for patients. And so then Derek will dive into the data, a little bit what we've come out. I know we've given updates in the past that the trial has finished and early signs or early data look good. And now we're going to dig into a lot more detail into the study itself and some of the outputs, some of the immune responses that we were observing from the clinical study. So I'll pass that on to Derek, and then David will step in and give context. What this data means for POOLBEG, for ultimately for the industry, for patients and for pharma companies, because ultimately we end up partnering with pharma. So briefly, as a top line, let's call it overview, positive results from POOLB 001 LPS human challenge trial. So as you know, 001 is a small molecule modulator being developed to address unmet medical need arising from severe influenza and other acute inflammatory conditions. And in this situation, it's the immune system that goes awry. The immune system is causing damage. And now we have a drug, POOLB 001, that we feel will block that negative, that aberrant immune response, and bring the immune response back to baseline, as it were. So what we're seeing now from the outputs from our clinical trial is a highly significant reduction in P38 MAP kinase-driven cytokines. Obviously, the drug hits P38 MAP kinase, a master regulator of the immune response. So again, having this human data is very compelling, both for ourselves and for our partnering objectives. We see marked reduction in multiple markers of systemic and local inflammation. So again, very importantly, your whole body as a whole, the decrease in inflammatory responses, but then localized as well. We have a very good understanding of what's going on in a local setting. And again, there's a clear dose-response relationships. As you see, one of the objectives is to kind of understand what level of the drug we need to put in patients moving forward. So we've got a very much better understanding of that situation right now. Importantly, for this particular drug, it's well tolerated across doses and no serious adverse events. Again, very important that this drug works well in patients, but obviously they don't get any negative side effects that could cause issues for prescribing in the future. And now what we feel in hand with this data is a comprehensive data packet that can now be shared with potential partners. So again, we're very excited about bringing this forward. As I say, it's been a Herculean team effort to get all this kind of work done, the data ready for sharing, and very excited to say what the molecule is doing in a human setting. So what I'll do now, I'll pass it on to Derek. The overall strategies, Derek will kind of walk through some slides, some of the science, and David will talk. We'll give about maybe 20 minutes or so for that. But of course, there'll be time for questions at the end of these studies. So again, we see a strong potential for POOLB 001 to be a blockbuster treatment for severe influenza and looking at other inflammatory conditions as well. So again, appreciate your time this morning. And now Derek will go through greater detail for the data. And as I say, just to talk about the two arms that we're looking at here, the severe influenza aspect is very exciting for us, and as we mentioned recently, kind of building into oncology as well, and we could touch upon that. We see this drug as a master inhibitor, let's say, of the immune response, as roles to play in severe influenza and oncology, and of course beyond. So again, very exciting time for the company, and I'll hand it across to Derek now to go through our data slides.
Thanks, Jeremy. And colleagues, I'm in central London, so if you hear sirens or alarms, please bear with me. So I'm a basic scientist. I'll be providing advice to Jeremy and colleagues at POOLBEG about how to determine the efficacy of POOLB 001. I'll refer to it as 001 from here on in. And I think one of the sort of things that to get in the background is inflammation is a good thing. And as part of the inflammatory response, we feel sometimes lethargic, high temperature, we feel achy. And these are all good things because it means our immune system has engaged. It's responded to the infection, the virus, for example. And all of these symptoms and effects are driven initially by what we call cytokines, the cytokine storm. And these are important molecules that prime the immune system to work efficiently. And we experience it every year when we get a flu or a cold. And certainly with COVID-19, we've learned a lot about the cytokine storm, and we've heard a great deal about how it suddenly becomes out of control, misbehaves, errant in many ways, and also as a side effect of some CAR T-cell therapy. We don't know really why a pathogen or infection does this to the immune system, but we certainly know the pathways that mediate it. And nature has been very good to us. It's given us many of these master regulatory pathways that Jeremy mentioned earlier on that ignites the flames of inflammation. And one of these is P38 MAP kinase. It's everywhere in our body, located primarily in the immune system, however. And it takes these pathogens, it tells our immune system that they're there, and it ignites the immune response. And if all is well, inflammation switches off. The problem, and we really don't know why this is the case, is why doesn't the inflammatory response switch off? Why does it persist? Why does the cytokine storm persist? And why do all of the bad things that cytokine storm does, why do they continue? And we know that there's immune cell activation. We know these immune cells leave the blood, get into the tissue where the infection is, primarily lung, in the case of flu. And then this becomes even more systemic, leading to organ failure, and in some cases, of course, death. So the P38 MAP kinase is very much front and central in driving and perpetuating the cytokine storm and its inflammatory sequelae, as you want, if you wish. And so POOLB001 was designed to block the behavior, the immune and biochemical activities of P38 MAP kinase in the immune system. And so we set about asking the question in a human experimental model that captures all of the essence of the inflammatory response, to ask the question whether 001, to what extent it can block these pathways and how many of these pathways it can block. And in collaboration with CHDR in Holland, a group of individuals aged 18 to 55, I believe, were recruited into a trial. And the trial had two arms, if you wish. One was the injection of a inflammagen, as we would call it, a bacterial product, locally into tissues, into the skin, using skin as a window into human immunity, if you wish. And this provides information on how the inflammation in humans evolves and switches off. And the second part of the study was injecting the bacterial product, again, endotoxin, IV, to capture the systemic response. And the reason why there is two arms is that injecting the bacterial product into skin is a very localized response. It doesn't spill out into the blood system. Hence the second study where the endotoxin or the inflammagen was injected IV to capture the systemic nature of an inflammatory response. And 001 was dosed a day or two before the inflammagens were injected into the individuals. And the reason to do that beforehand is that we know a great deal about these models. We know how they behave in terms of the cytokines that are released. We know when they are released. We know where to find them. We roughly know what levels they are and at what time points during the inflammatory response. And we also know the impact that these cytokines have on blood pressure, body temperature, CRP, which is a very recognized clinical marker of well-being. And so by dosing beforehand, we can ask the question, as I said earlier, how many of these inflammatory events are being blocked by 001 and to what extent they are being blocked. So it's a very scientific approach to the problem. And the readouts are local measurements of inflammation. We can image the skin. We can see how red it is as a consequence of injecting the inflammagen. We can take the inflammatory fluid out of the site of injection and measure all of the inflammatory cytokines and things we know are important for inflammation and things we know drive a very severe overexuberant acute inflammation. And there's also skin punch biopsies, which are incredibly useful and informative to immunologists like myself. And it can also inform the study on safety and tolerability, which, as Jeremy said earlier on, is very well tolerated. And again, the IV injection allows us to capture, as I mentioned earlier, all of the systemic markers of errant overexuberant inflammation. And for me, as an immunologist or an immunopharmacologist, this slide tells us something really important, that giving 001 to healthy individuals gets the drug into the body in a dose-dependent manner. It tells us how much of the drug is there after dosing orally. It tells us how long the drug lasts for. And importantly, it tells us that this occurs in a dose-dependent manner in the body of these individuals. And my next slide is asking the very nerdy biochemical question, does 001 engage with the P38 MAP kinase pathway, this master regulator, as Jeremy said, of the acute inflammatory response? And the slide on the left shows you that really quite nicely. And the gray bar or the gray line shows what happens when we inject LPS, which is a shorter term for endotoxin, into healthy people. It does exactly as it says on the tin. P38 MAP kinase becomes activated. And as biochemists, we measure activation by measuring phosphorylation, the addition of phosphate groups to P38 MAP kinase. We love a good phosphate group in immunology. These days, in the gray bar tells us that the immune model has reacted and P38 has reacted as we'd expect within 1 hour of injecting the LPS. And with 001 on board, we can see a resounding and very convincing inhibition of the phosphorylation, or if you wish, activation of the P38 MAP kinase, this key pathway we want to bring under control during the cytokine storm. Now, there are many readouts then of this biochemical inhibition of our key target. I'm choosing TNF-alpha here simply because it is one of the first cytokines to be released when the body sees an infection. Secondly, it's particularly well studied in the field of systemic inflammation. Thirdly, it's present at very high levels within blood. Fourthly, if you happen to inject TNF-alpha into people, you get all of the hallmarks of a cytokine storm, or if you wish, of a flu. But also TNF-alpha ignites other parts of the inflammatory response. And so you get this image of an inflammatory cascade. But again, that is elevated in response to the LPS, or the bacterial inflammagen, as we call it. And with 001 on board, you get this very dose-dependent and convincing inhibition of this TNF-alpha in the blood circulation of these individuals with this drug on board. So it tells us that the drug is getting there, it's engaging its target, and it's doing exactly as we would hope it would do. And as a consequence of blocking TNF-alpha and other cytokines and chemokines that we didn't mention, this also resulted in a reduction in the increase in body temperature. These are the healthy individuals, the placebo-controlled, did not get a drug, but they received the inflammagen LPS, and their body temperature rose somewhat. But with the drug on board, this impaired the increase in body temperature. So the increase in body heat that you get when you get a flu is being blunted. And in line with that is also a dose-dependent decrease in heart rate that is caused by the injection of LPS. This is depicted in the gray line. Our heart rate increases, as you'd expect in the healthy people, given the bacterial product. But in green and in red and blue, then the drug dose-dependently reduces heart rate, telling us again that the inflammatory sequelae of the cytokine storm is also being blunted. And we all recognize C-reactive protein. This is a systemic marker of human inflammation. And again, that's dose-dependently and significantly inhibited, or resoundingly inhibited, with 001 on board. So all of the hallmarks of a systemic inflammatory response caused by the inflammagen LPS or endotoxin have been, I think, convincingly blunted and inhibited by 001. That's in the blood. Moving on to the next slide, second last slide for me, is asking what happens in tissues. I told you that we did 2 arms. We injected the LPS or the inflammagen IV for the systemic arm, but also in tissues, because we know that's in fact where a very severe infection starts, often in lung or in the peritoneal cavity, and then disseminates. We also looked into the skin, as I say here, in localized inflammation. And again, if we look at our favorite cytokine, TNF-alpha, levels elevate incredibly quickly within the tissue, in the gray bar, as you would expect. And these have been dose-dependently blunted with 001 on board. Again, telling us that not alone did 001 get into blood, but it also managed to get into tissue. And in fact, it's in tissue where a great deal of the inflammation-driven harm has occurred. And a lot of that inflammation-driven harm is brought about by the overexuberant infiltration of what we call granulocytes or white blood cells, neutrophils, but also monocytes are a hallmark of this type of event. And I'm not showing the data here today, but in as much as TNF-alpha levels were reduced at the site of inflammation, the numbers of white blood damaging neutrophils and monocytes were significantly and very impressively inhibited in the tissue in response to 001. So I also like to finish off with something very visual. I think for me, this captures the efficacy of 001. That's not Jupiter, Mars, and the moon. That is an image of the skin of the individual volunteers injected with the inflammagen endotoxin of LPS in the placebo-controlled people. This gives a sense of the localized inflammation. It's like an exaggerated bee sting, if you wish. And this shows you how effective visually that 001 has been in quelling or dampening that localized inflammation. So not just trying to convince you that within the tissue, these invisible cytokines, chemokines, hormones are blunted, cells are also reduced to number, but visually you can clearly see how effective the 001 is. So in summary for me, 001 gets into the blood, it engages with its target, this master regulator of inflammation, P38 MAP kinase, that becomes overexpressed and overactive in very severe pathogenic infections like COVID. And it reduces in blood the key chemokines and the cytokines and markers of alarm bells ringing of inflammation about to kick off and do very bad things. It also gets into tissues, very important, blocks the chemokines, cytokines, and the infiltration of potentially tissue-damaging white blood cells. And of course, then, as I said, it dampens or blunts the heart rate, body temperature, and CRP levels. And that's it for me, Jeremy.
Thank you. Thank you for that, Derek. I love a bit of science in the morning. I think it's a nice overview, and I will say this is a kind of a snapshot of the data set that we have. What we have is obviously very much more kind of comprehensive, but we're delighted what we're seeing. Again, appreciate Derek giving us that overview of the key components, as I say, which kind of convinces us now that we have a drug to move forward with, as he said, binding, engaging the target and blocking those inflammatory responses, which we see has great potential in severe influenza and beyond. But obviously we want to put this now, the science is now, as I say, at this stage. Let's talk further about putting it in context from a POOLBEG standpoint, and David will speak about realizing value through business development, the ultimate strategy for the company itself. So over to you, David.
Yeah, thanks, Jeremy. Thanks, Derek. I think I'll talk about 3 things today. The first is influenza as a space. So some broad numbers for those listening to just contextualize what the market currently looks like, what the treatment options are, and what specifically the standard of care is for severe influenza. The second piece is I wanted to cover an aspect of our operating model around partnering and specifically why we partner when we partner. And then finally, I was going to chat a little bit about the out-licensing piece. I have, I think it's 6 slides. So I'll try and be brief on each slide, maybe 45 seconds to a minute, but the goal again is to leave time at the end for Q&A, and we can jump back in as needed. So I'll start moving through the deck as it stands. So some of the basics here. So influenza broadly, this is a slide, and much of this data is from WHO. It's just framing the overall scale of the issue. So there's 1 billion cases annually for general influenza, 5 to 10 million hospitalizations based on WHO, and there's about 650,000 respiratory deaths per annum due to influenza itself. It impacts 30% of all children, 10% of all adults. What's worrying, of course, in addition to that, is that if you have influenza, you're 6 times more risk of getting a heart attack, 8 times more at more risk of getting pneumonia, and 8 times risk increase in terms of stroke. And if you go beyond the treatment per se, there is a significant economic burden to influenza, particularly hospitalized patients. And the estimate at the bottom of the screen shows the economic burden around $11 billion. That number is a number of years old, so it could be even higher than highlighted there. So let's talk about what the marketplace looks like right now. So influenza has been around obviously for some time, and the market is led by Tamiflu. So people are well aware of that brand, and it's about a $2 billion market, general influenza. Tamiflu is about half of that, about $1 billion. But what's interesting with these kind of antiviral approaches is that they're approved for acute, uncomplicated influenza. And specifically, what they do is block viral replication, and that's fine in early disease. But as disease becomes more complex, these therapeutics aren't particularly effective, and to that end, aren't FDA approved for more severe forms of the disease. Again, it's not just the treatment piece. Holistically, when we think about the healthcare system, there are quotes here from the Gates Foundation and the National Institute of Health in the US, highlighting the fact that there's a major unmet need here that's not being effectively managed, and there's no adequate treatment for patients who have severe symptoms and are hospitalized. Let's speak then to severe influenza itself. Again, the numbers, it's about 3 to 5 million cases globally per the WHO. That may be underreported. And the analog is of around 650,000 deaths per annum. And with a lot of these patients, we are seeing that severe cytokine storm. So what happens in practice is these patients come into a hospital with malaise and a lower respiratory tract infection. They're initially put into the high dependency unit of a hospital, and then as they progress, they may be moved to the ICU. There are a number of risk factors that drive movement towards severe influenza: respiratory challenges, heart disease, obesity, and even pregnancy. In terms of current treatment, there is no approved medication for severe influenza, and the focus is predominantly on symptom minimization. So what happens in practice is that they're primarily given long-duration antivirals post-hospital admission. There is guidelines to state that, but the drugs out there have been proved ineffective to date. And then beyond that, they are put on treatments to try and oxygenate the blood to prevent mortality. No currently approved drug, and a significant need exists to A, address the unmet need, and B, have a more targeted therapeutic that can really resolve the issues, both from a patient perspective, but more holistically for the healthcare ecosystem. Jeremy mentioned briefly, we're not just looking at severe influenza, we're also considering additional indications or therapeutic categories beyond influenza. So that might be adjacent indications to influenza, or something quite different, such as oncology. And specifically, we're looking at the CAR-T space as something where we can address the associated toxicities we're seeing with hyperinflammation with these patients. We can certainly speak to that on the Q&A or another call to go into the CAR-T space in more detail. But I think as a business, it shows we want to fully liberate the value of the molecule. We want to really think about multiple indications where this novel therapeutic can be prescribed and used. One question I think then that comes up a lot is the fact that we look to partner early. And when I talk about partnering early, I tend to talk about pre-phase 2. Historically, we would have seen within the pharmaceutical industry, it was more later stage partnering, which made most sense for big cap pharma companies. There's some data on the top, on the left panel, I should say, from JP Morgan, that's showing a change in trend over the last decade or so. And specifically what we're seeing is that bigger companies are leaning in or getting involved earlier in licensing deals. And specifically, I guess the bumper sticker is that we see just under 90% of 2022 in licensing deals were pre-phase 2. So we think it makes a lot of sense to de-risk the molecule, partner pre-phase 2, and then work with parties who essentially have complementary skill sets to accelerate the science and bring it to market faster, given that we're a relatively small biotech. So this is a positive thing to see, and it aligns with our thinking as we go to market with this program. Final slide, just talking about kind of out licensing efforts. I would say, what are we grounded in first? I think we're grounded in an operating model that's scalable and sustainable. We're talking only about one asset today, but we're certainly not a one asset company. We have multiple programs in development, and we're looking to move those along in due course, with this program obviously being our lead one. When I think about what we do at a simple level, I think we try and focus on problems we can uniquely solve ourselves, focus on problems where there's a clear unmet need or ineffective standard of care. Subsequently, there will be a clear market potential. And then we look to build meaningful early data, such as our preclinical work, our phase 1 work, and our LPS work with 001. And then ultimately, we partner with larger companies downstream to accelerate these novel medicines to market. When we talk specifically around 001, what's exciting to me about it, a couple of key points. We've positive phase 1 and LPS challenge studies are now completed. We're patent protected until 2038. This molecule is agnostic to viral strains, so it's almost like an anti-inflammatory response to the condition rather than an antiviral. There's currently no approved therapy in this market. So if you assume 5 to 10 million hospitalizations, there's no adequate treatment there. And I think we've got the potentiality then to address other acute inflammatory diseases beyond severe influenza. And we're actively looking at that, of course, to liberate the true value of the molecule as such. In terms of then partnering, as mentioned previously, we will look to partner pre-phase 2. And we're looking for those mutually supportive partnerships that can advance the science and deliver meaningful solutions. We're thinking about both global or regional deals, and we are already engaged in discussions with interested parties. So we've been keeping bigger companies abreast of our developments over the last year or so. And we're looking forward to a constructive 2023 in lieu of further discussions with companies on the program. So, Jeremy, I think I've covered the majority of the items on the slides, and obviously we can take Q&A if you'd like to address anything in more specific detail.
Yeah, appreciate that, David, for giving us the overview of the context. I guess here we are. We're in an exciting phase with the company, with the early human clinical data with POOLBEG 001, which we see can be broadly applicable to severe influenza and beyond. Again, very exciting time for the company to have this data in hand and know that we have a drug now that works. It hits the target, as Derek said, it blocks that inflammatory response, and now it's a case of taking it into the next level. So again, very exciting time for the company. Again, my kind of appreciation to the team behind us here at POOLBEG, who've worked tremendously hard to get us to this point, and say it's an exciting time for the company. But David said, what David said is correct. We have other programs, coming through as well, or artificial intelligence programs, oral GLP-1, etc. We can talk about at another time, but today's focus is on POOLBEG 001. So again, exciting time for the company. Yes, there's work to do moving this forward, getting partnerships and generating that revenue from the partnership scale. So again, fantastic time for the company. So what we'll do now is we'll transition over to some Q&A. I see there's some questions coming in. What I do, I'll read out the questions, and then between the three of us, between David, Derek, and myself, we'll give some answers best possible. If we don't have the answers today, obviously we're happy to follow up with greater specifics. So we'll kick off from Robin D. Can you expand on the dose-response relationship? Just a heads up, Derek, this one's coming to you. How does the LPS challenge model translate to the real-world clinical setting for influenza, etc.? And can we assume that you've established an RP2D, that's your recommended phase 2 dose? So, Derek, do you want to comment on that?
I can certainly speak to the first two, and we can trash out the third part of that question between us. Yep. So the dose response, I'm quite sure the nature of the question, but I guess 3 doses were chosen, 30, 70, and 150. And the nature of that dose response was that as the drug increased in blood and engaged with its target, that we got a corresponding inhibition of the ensuing inflammation. And in pharmacology or immunopharmacology, it's a real test that a drug is doing what it says it will do. As you increase the concentration and engages with its target, you then get a corresponding yang of the inhibition of the target outputs. How does this model translate to the real world? What this model does is it captures every aspect of an aggressive inflammatory response that you would see with an influenza infection. It captures all of the chemokines and the cytokines, all of the white blood cell misbehavior. It captures the heart rate. It captures body temperature. It captures CRP. So it's a controlled experimental setting to ask if, in principle, the drug works, and it does. The window of P38 MAP kinase-driven cytokine storm and inflammatory sequelae in the real world will, I suspect, be much wider, in other words, be more protracted. So in reality, I would suspect it gives 001 a greater opportunity to engage with the target and then block many, if not all, of the inflammatory events driven by P38 in an infectious disease setting. So it's a controlled way of verifying that the drug works, and it indicates to us that the drug will blunt, dampen, inhibit all of the acute inflammatory cytokines in the flu setting.
I was just going to add, Jeremy, onto that around the recommended dose. I think it's clear for us internally we have our sense of where we think that might be, but given the fact that we are looking to partner pre-phase 2, it would be a conversation with any partner. So if we do a licensing deal, ultimately their team will take a look at it and make the decision downstream. But certainly I think we've reviewed the data robustly and have a good sense of where we think things would lie. So I think it would be a little unfair for us to comment specifically on which dose we think that is.
Yeah, it's a good point. Since partnering is our main objective, you're right. That'll be a conversation with the partner. That being said, I know talking to our chief medical officer, we looked at the 70 mg data and the 150 mg data. They're actually quite similar. The 150, there's some kind of advantages, and we know that safety is confirmed. So there might be that window. But as I said, these discussions are occurring internally. But you're right, David. These would be more kind of external discussions with partners. But 70 or 150, that's a good discussion to have. We know we're in that window. So I think that's the positive that comes from this data set. So again, Robin, appreciate the question there. Question from Nathan Orr. How much conversation has been had around accelerating POOLB 001 for bacterial sepsis, especially gram-negative organisms such as E. coli or Klebsiella, common cause of sepsis derived from the gut or urinary tract? Very good question. I think, I'm 20 years now in the industry. I think there's a lot of questions about let's, take programs forward, get that early human data as we have done, get efficacy data in larger clinical trials where we know it'll work, and then go to the more complicated diseases. Now we know that sepsis is significant unmet medical need, but a very, very challenging disease to pursue. As you said, like there's a lot of issues out there with it for multi-drug resistance, even beyond that. So I think starting out in the severe influenza setting and then branching beyond there. But again, to David's point, that's a conversation with pharma people that, if they want to go into these more complicated trials, that's a direction they can take. And they know clearly there's an unmet need here that potentially POOLB 001 can play a role.
Absolutely. I would agree with that, Jeremy. I think maybe I mentioned in our previous slides, we're looking across a number of indications. We obviously couldn't state publicly what they would be until we generated IP, right, or a partner did. So we're always going to be cautious on that, but I wouldn't mistake that caution for a lack of rigor in terms of our analysis. I think we've done and are doing a good job on that front.
Yeah, appreciate that, David. Next question, David, I'll give to you. I think it's answered or partially answered, but what's next for POOL001? Are you funded to take into phase 2 or will look to partner? I think you addressed that, but if you want to kind of add.
Yeah, absolutely. I think it all goes back to our operating model, right? I think what we do is we target problems we think you can uniquely solve. So from an in-licensing perspective, we have a team that assess those. We look to then further de-risk the molecule with early meaningful data. And then what we bring to pharma is a kind of a de-risk program, right? If you look at the probability of success of drugs over the last decade across every indication, they really haven't changed. So what we're actually doing is increasing the probability of success. So for any of the analysts on the call, that drives up your NPV of the asset, right? So that's what we're looking to do. In terms of our funding, I think we probably could fund the phase 2, but I think what we want to do is be that early stage engine for bigger companies, because ultimately they have bigger scale, right? They have large clinical teams, large market access teams, large commercial teams, and oftentimes will have clear opinions on how they want to bring the asset forward. So I think it's more around sticking to our operating model, which we think is smart and also scalable beyond 001. So it's more of a case of aligning with that. And as I said, I think the data on the JP Morgan study is quite compelling. Companies are more eager to get involved early. I know in historical companies I've worked for, we've got involved sometimes at phase 2 or phase 1 and gone back to preclinical to reassess the optionality or the potential of the molecule. So I think when companies go in and are attracted to a given asset, they want to give their own slant on it, their own take on it as well. So I think that's why we're partnering at that stage.
That's a good point. I will add as well, and David, you touched upon this, but when we have, let's say we have thought about what a phase 2A would look like, what a phase 2B would look like, costed or budgeted those up, have an understanding. So when it comes to pharma discussions, we see kind of deals could be a straight clean out license. As David said, it could be a territorial deal, or it could be kind of a development where they would fund it and ask us to run it, etc. Just, so there's different. So again, optionality is David's words. So there's that option that we've pieces in place from a deal standpoint. We can take it forward in multiple directions. But again, Niall, I appreciate that question. Moving on, Robin's in with another question. I think I'll take this one myself, but is there any further R&D work that Poolbeg might consider on the POOL001 program, perhaps short of a further study, that can be done while you're seeking a licensing partner? It's a reasonable question. I think the one point to point to is, we are looking at other indications. We are looking at other diseases. We mentioned, that CAR-T is a direction we can take. So we have kind of thoughts and plans on to move that forward. And again, building the business case that this is a molecule. We've mentioned, I think all three of us that, as a master regulator, we know that this can go in many different branches, let's say, of immunology. Many different diseases can be treated with 001. So we are exploring different directions there. So again, appreciate the question, Robin. A question from Mark P. Actually, we may need the slides back up for this. Derek's final slide, the 4 lines need explanation. The vertical ordering looks surprising to a layman. Okay, let's jump back here. Okay.
Excuse me. Righty. So I put this in simply because I wanted to illustrate visually the local effects of 001 on the inflammation. The grey line is the degree of redness, or as we call it, hypervascular reactivity, how the little blood vessels around the site of infection have expanded to deliver immune cells into the tissue. And that's the typical redness that you get when you get sunburn or a bee sting. And predictably, that peaks at around 3 to 6 hours. And it lasts for actually up to 24 hours. And we've done this many times ourselves, as well as CHGR. And after 24 hours, it comes down as the redness and pain, heat and swelling of a bee sting would do. What these data show is that the drug 001 is very good, actually, blocking the redness and the vascular hyperreactivity. But it loses its efficacy as time goes on, and this is completely in line with the pharmacokinetics and the half-life of the drug, such that by up to 24 hours, or if you like, between 9 and 24 hours, the drug has gone about 75% of it, 80% in the blood, and therefore it's lost its efficacy of blocking the redness. Now, there's an old thing in immunopharmacology called the rebound effect, and it's always been seen in studies where early anti-inflammatories were given, particularly to rodent studies. And these drugs are very good at blocking the early onset phase in response to an infection. But when the drug wore off, then the inflammation rebounded. We don't see evidence of that here. And so the drug has managed to block the inflammation and then keep the ensuing inflammatory response under control. So it's another, for me, a very interesting aspect of the immunopharmacology of this pathway and of 001.
Super. Thank you, Derek, for that explanation. Moving on, Adam B. We've seen some drugs like Tamiflu where getting the drug very early does influence efficacy. Absolutely correct. Within 48 hours is the window on the label. Is there a time frame effect here? I.e., do you expect the drug will need to be administered within a certain time frame from symptom onset? And is there any prophylactic angle here? Really good question. So when we were kind of progressing through with severe influenza, now these are patients who obviously have the virus. You know, most people get mild to moderate and recover. You know, you're feeling terrible for several days to a week. But it's the people that progress, that actually become hospitalized, that are, you know, ending up in the ICU. This is where, again, their immune system has gone too far. The cytokine storm is kicking in. So that's what we're aiming at to begin with. These are patients that, as I say, they need more help than anybody. And as David mentioned, there's nothing else out there for them. So that's the starting point for us. But you're correct. I mean, you could appreciate there may be a prophylactic angle that the earlier you go in the setting, that you know, the drug may have better benefits. But I think from a drug approval standpoint, you know, you want to get into these sicker patients, prove efficacy there, and then work your way backwards. So it's a really good question. And Derek, please.
Another bit to add to that is that I think we didn't touch upon it, but this pathway becomes overexpressed, or it becomes upregulated as we get older. So as we hit 50, 60, 70, and 80 years of age, P38 MAP kinase just naturally, as a consequence of years of evolution, has just increases in our immune system. We don't really know why. And the rub of that is, as you get older, you get more P38. As you get infections, you'll therefore more likely get a more severe cytokine storm. And we can remember from COVID-19 that it was primarily older people who, when they got COVID-19 and got the inflammation, died. And so there will be a real prophylactic case of older people who do get infections and who are showing signs of cytokine storm to get this drug on board early. So it'd be very easy to triage and identify the individuals who will benefit from this drug.
Yeah, good point.
Jeremy,
yeah, David.
I was just going to add to it. I think we covered it earlier when we said, like, our focus is in acute inflammatory conditions, right? So certainly, as a starting point, we'll be looking at hospitalized patients that are brought in for treatment, because that's kind of where severe influenza is diagnosed. Not to say that it doesn't happen earlier, but that's just the way the healthcare ecosystem works.
Yeah.
When I think about it then from a valuation perspective, one of the reasons we go for a very distinct population and show data in that is that data drives pricing.
Yeah.
We're not looking to compete with generics. We're not, our low-cost antivirals. We're going for a pricing that is reflective of the unmet need we're addressing.
Yeah.
And then beyond that, certainly over time, from a life cycle management perspective, you could look at going maybe earlier. The scientific angle of that is beyond my expertise, but generally speaking, it goes back to distinct populations with a clear market potential, and then work thereafter. And certainly if you talk to US payers, I would have done a lot of research with US payers in my last job. You need to show clear clinical outcomes to ensure that, one, you're on formulary, and secondly, that your pricing is reflective of the R&D investment that has gone into the therapeutic.
No, you're absolutely right.
I think it's that's the starting point, as you said. The FDA label will be maybe in that area, and then we can move thereafter.
No, that's a very good point. As I say, the kind of selection and the pricing that drives on that is very important. Actually, to Derek's point, it reminds me of conversations I've had with our good friend Professor Luke O'Neill, who sits on our board on SAB, about the inflammaging And it all reminds the fact, if you look at life expectancy over the years and the centuries, about 100, 120 years ago, life expectancy was about 55 years. So now we're pushing 85, 86, 87. So from an evolution standpoint, the immune system doesn't know what's going on because it's not used to these scenarios. But obviously, this is where therapies or therapeutics can kick in as well, just to kind of control these master regulators like P38 with 001. All right, a question from Alan D. This one's for you, David. Will your partner licensing strategy be indication specific in flu or oncology, etc., or will you look for a single partner to license all rights and geographies for the right price, of course, to POOLB 001?
Yeah, I kind of have the politician answer on that, and it depends, right? So we have to be open to kind of hearing the other side and what they're looking for. Certainly one avenue, as the person mentioned, is we could do a global strategic deal with a big company who have therapeutic expertise across a lot of indications, and they would fully out-license it for all those indications. That's typically what you see. We could also look at a regional deal for APAC, let's say, or Latin America, and split it up that way. In terms of like siloed indications, that's less common. Particularly if there's overlap in the call points from a Salesforce perspective. So it just really depends on the case-by-case basis. Mm-hmm. But I would say we're not shutting out any options. We're available to listen, and we talk through with these companies. And ultimately, it's a two-way conversation to get to the valuation of the molecule, right? We need to be able to have a shared agreement on the forecast, what the NPV is, and ultimately then we talk about splitting the pie thereafter.
I think that makes perfect sense, keeping, as you say, optionality. Yeah. Moving on, appreciate that question. Again, thanks very much for these lots of questions coming in. John S. Okay, this is a multi-part question. I'll read it out and then maybe we'll break it down from there. But is POOLB 001 selective for any specific P38 forms, such as alpha? Good question. Is this a novel molecule as patent till 2038? And what are POOLBEG royalties back to Discovery Partner? We'll talk about that. On markets that are are there any specific US market flu figures? And how long do you see POOLB 001 being administered, as P38 inhibitors historically fade after the first week? Okay, there's 4 questions there. All right. The P38 forms, yes, I mean, it's a cross spectrum. I think it's alpha, beta, if I understand correctly. And again, hitting the key isoforms of P38 in that, from a novel molecule standpoint, and David has done a lot of work with it. David, do you want to comment on the novel molecule and the
on the IP? Yes, sure. I guess people will have known that P38 was used in the past for more chronic conditions, chronic indications, and Big Pharma had an interest in that. Some went through the clinic to maybe phase 2 or 3 and didn't move forward, particularly because there was toxicities associated with long-term use, so months of use of therapy. What we're doing is we're looking at acute treatment, and we're not seeing any of these. We've obfuscated the tox side effects. So I think the patents we have are method of treatment, but what's unusual here is that because a P38 has never gone to market, we should be able to get NCE status in the US, so market exclusivity in the US. In addition, we should probably be able to get additional market exclusivity in the EMA region. So that's typically an 8 + 2 + 1 in terms of years. So I think it's a sort of an unusual approach there. And I think that's really compelling to bigger companies because we kind of have a clear understanding of our IP and why we're protected within this space.
It's a good point. And for those playing acronym bingo, NME, new molecular entity. So as David said, P38s had been experimented in more chronic diseases like rheumatoid arthritis, didn't work out because of that long-term treatment. As a distinguishing factor, we're looking at these acute conditions. So we know we're only going to give 1 or 2 weeks treatment. And I think that goes to the point here about how long does POOLB 001 be administered. We see this as, again, if you think about the setting where patients are coming in, they're in a prone position, they're in severe influenza stage, they'll be in the ICU. So 1 to 2 weeks, treatment. And again, it'll be kind of daily dosing. The study we did here is twice daily. So, we'll understand that it goes back to the phase 2, understanding the phase 2 dose. So it will be kind of, again, likely an IV form as well. So there's many ways we can push this forward. So we do have a good understanding. I guess the last part of that question, the US flu market specific, I know we have global numbers, but I think there are breakdowns there as well. And as you know, the US are, they're there's a big push every winter to get in their vaccines to the elderly in particular. So I think we do have those numbers, and again,
yeah, we do have those, and I mean they're readily available as well on the CDC website and WHO. I think the numbers we shared are, of course, global numbers, but they are relatively up to date.
Yeah.
They are readily available, and I think we've shared them on older slide decks, but we can certainly update them with this deck if you'd like, whatever.
Yeah, that's a good point. All right. Yeah. A question again from Alvin B. Maybe we don't have the right people on the call, so this might be one to follow up. But is there a chance regulators would accept human challenge data instead of a phase 2 study? I can comment on that. Or would you anticipate having to perform a phase 2 trial as opposed to potentially moving straight to phase 3? It's a really good question, and I see where the question is coming from, because colleagues at hVivo have ran viral challenge studies, so human viral challenge studies, where you're deliberately infecting volunteers with a virus and then testing antivirals or testing vaccines, given previously administered. So I think there has been examples, particularly in RSV, where they were able to take this phase 2 data, you could call it, to the regulators and get permission. I think from us, as I said, that this LPS study gives us the kind of baseline. We still need to put it into patients. It could be conceivable that yes, you could do this in a flu setting. But if you remember, we're going after the severe influenza settings. These are patients who are really quite sick. So in the challenge, clinical trial setting, these are patients who do get the mild to moderate and they recover. They're usually younger, etc. So we're going for a different population. But it's an interesting question. Again, a discussion with pharma partners. We know that the likes of, let's say, Pfizer, for example, have done a number of these viral challenge studies, particularly in RSV and indeed in flu. So they'd have a good understanding of that regulatory path. But as I say, a good question and good thoughts. So
yeah, I was going to add a little bit to that, Jeremy. Certainly, I would say the first thing is that we've de-risked the molecule to some extent, right? We have a preclinical done successful, phase 1 done successful, LPS successful. My expectation that is quite likely a partner will have to do a phase 2 and phase 3. That can be different, of course, than if it's an oncology, right? You see that sometimes the jumping from a phase 1 to a pivotal. But I would expect that a phase 2 would need to be done, and we've done the homework on that already. And when we discuss with partners, we're giving them our take on what it might look like. So I would expect a phase 2 to be required for sure.
Yeah, no, absolutely on that. Super, David. All right, thanks for that. Just looking through, I think we're running out of questions, which is, again, appreciate that. Let me see if there's any new ones that have come through. Charlie A, what level of phase 2-3 trials are likely to be needed for 001, given the history of the drug? I'm not too sure what you mean by level phase 2-3 trials. I mean, we touched upon it that we have a sense or an understanding of the trial, the numbers of patients required. Let's say when it comes to a phase 2A, phase 2B, the overall cost of what they will be, but these proof of concepts. Phase 3s obviously they kind of scale up in multiple territories. And that's where certainly that's where our pharma partners will be looking for. So again, it's in that discussion. There are examples out there of clinical trials in the flu setting, but not necessarily the severe influenza setting. So again, we have a lot of thoughts going on in the background on that. So we can update as we progress, as I say. But I think that's more for partnering discussions than Poolbeg ourselves moving forward. Again, appreciate the question. From Sukman G. Sorry, he's disappeared. He was there a second ago. Sorry. It's actually my computer's acting quirky here. From your partner discussions pre today's news, did you tick all the boxes on what they would want to see from the data before signing a deal? I think, David, we've had good discussions with pharma. I've always said from the beginning of Poolbeg, this early human efficacy data is really what gets them excited, enables them to go to management and senior management with a comprehensive data set, and then designs to move them forward from there. But so some discussions. As I say, this is I think a key inflection point for both our discussions and Poolbeg in particular. But again, David, if you want to comment on that. And
I obviously couldn't go into specifics, but I think we have a compelling story and people understand A, the approach, B, why we've taken the approach, and C, our thoughts on what next steps look like. So I think they've been constructive discussions.
Yeah, no, absolutely. But this is a key inflection point, having this data in hand.
Yeah.
And this is what they're waiting for as well. Sorry, Sukman's questions has popped back up. Great work, well done to all. Appreciate that. Thank you. do you intend to dual list POOLBEG possibly on NASDAQ? We've mentioned this before, the fact that, we have ambitions beyond the EA market. We see the US market as being very attractive. I think there's a lot of interest. And again, we've mentioned in the past where we continually have the discussions with companies over there about, the possibility of it. We have not lost focus. I think that's the key thing. We're very much focused on delivering from our pipeline and executing there. But yes, there are conversations that go on in parallel. But it's a case of finding the right time, the right valuation, the right company, etc. If you do a reverse merger, so that's all on the table. So as I say, there's we'll update the markets if there's significant progression there. Can you please comment on what you think a phase 2 study looks like? Size, patient characteristics, endpoints, duration. Again, probably not appropriate to go into that kind of detail. We can kind of follow up on that, but we have thoughts to say on a 2A, which is kind of a smaller trial in these severe influenza patients, maybe 15, 18 patients, and obviously that'll expand beyond that to 100 more in multiple sites when we look for a 2B. So these are kind of like your kind of next step studies of again, getting that next stage of proof of concept in a disease setting. We know the drug works, as we've commented on earlier on. We know that it binds to the target. We know it inhibits the inflammation. Again, fantastic milestone to cross with this data in hand. And then you can start designing what the endpoints will look like. So that's kind of a conversation for another day, let's say. But appreciate that. I think now we're coming up to close to the hour, so maybe we'll take one or two more questions from Charlie A, very active. Oh, sorry, he's clarifying. I meant, would phase 3 be needed as known drug with lots of trials? Okay, could you get orphan drug, expedited drug development from FDA as no current applicable drug for clearly addressable market? Again, I'd love to have our colleague Bruno Speeder on the call from a regulatory standpoint. But yes, I mean, these are possibilities, but I think he obviously steps with phase 2A, phase 2B in the interim.
Yeah, I would just add, Jeremy, I guess orphan drug is driven by patient epidemiology size. So this market would be too large for orphan per se. There is always the possibility that we could get fast track designation from the FDA. And I do think a phase 3 would definitively be needed. Yeah, so that'd be my, again, non-informed, non-regulatory person.
Yeah, Bruno's the man for that. But obviously, yes. I mean, Bruno, who maybe we'll bring Bruno on one of our other presentations to talk about, but he's very informed, very knowledgeable, all things FDA and EMA and what's needed to move things forward. So that's a conversation I think we can have separately. But really good question. Appreciate that. Obviously, the quickest to market is the approach that we'd all like to take, get it to patients as quickly as possible, and we can achieve that. All right, I see two more questions just to kind of coming in. Do you have PK data for the doses tested from work done by the prior licensee? Can we make assumptions about whether a single dose might be required or whether multiple doses might be administered over several days while the infection resolves? All right, good question. I'll start, Derek, and then maybe you want to comment on that. But we did mention this drug had been in phase 1 healthy volunteer trial, single ascending dose, multiple ascending dose, and so we have all of that data set. So we do have a very good understanding of the molecule, the kind of the ADME, the administration, metabolism, excretion, distribution, etc. We have all of that information. We do know the half-life, and again, we learn more from our 1B trial in an inflammatory setting. So I think that resolving on a single dose is unlikely. Derek can comment, but I think when we look at kind of these very sick patients in an IV setting, it's kind of a continual administration where they're getting this dose, because you do want to block B38 and just kind of calm it down, relax it, and then let the patient kind of naturally recover. So I think it's it wouldn't be a single dose, let's say, as a pill, but we do see kind of a 1 to 2 week treatment in this intensive care setting for the severe influenza. I don't know, Derek, do you want to add?
Yeah, I would have thought that would be about right. 10 days, 14 days, and of course, in the intensive care setting, all of these markers can be easily monitored. So when the drug starts taking effect, you can see that pretty quickly, and then you make a judicious decision as to whether to stop administering the drug or continuing. But my guess, and this is the quirky thing, the last 30 seconds, infectious diseases are cleared out of your immune system. So you're not worried about the infection. When 001 has been given, it's to dampen the errant, uncontrolled inflammation. So you're not really worried that if you dampen inflammation, that the infection comes back, because it's actually been long cleared. So I would suspect that once these symptoms begin to wane, as a consequence of the drug being on board, then that'll be around 10 days, 2 weeks, and then you can withdraw treatment.
Fabulous, fabulous. And on that note, I see that we're at the very top of the hour. Again, appreciate everybody for attending. Again, we are, as a team, delighted to be presenting the exciting data that we've generated through this LBS challenge trial. A lot of work to do. Very excited to get going. But I say this is a very important moment for the company and for patients as well. So again, appreciate your time this morning. If there's any, as I say, questions we didn't address, happy to do those after the fact. And again, thank you for your time, and we'll see you again soon with more Poolbeg updates.
Thank you.
That's fantastic. Thank you very much, Jeremy, David, Derek. Thank you indeed for updating investors today. Can I please ask investors not to close the session? It should be automatically redirected to provide your feedback in order the team can better understand your views and expectations. This will only take a few moments to complete and is greatly valued by the company. On behalf of the management team of POOLBEG Pharma PLC, we'd like to thank you for attending today's presentation. That concludes today's session. Thank you and
Appreciate
good morning to you all.
All right.
Thanks, everybody.